Okay, so the Echo 360 recording for, I think on Canvas it's listed as lecture notes 02B, lecture notes 03A, cuts off from an audio standpoint right in the middle. So I decided to go ahead and complete that lecture in a recording in my office. And we left that lecture off where the audio cut off. We left it off with this question. So let me come here with this question: where does the energy to make ATP in step 7 come from? And so we're going to pick up right there and look at some of these answers and then go from there into some of the answers that I like to help you understand with this, right? So, as you look through this, this one, a magic, it's magical, right? That's an idea here. We have breaking of bonds. We have electron transport chain here. Electron transport chain is not where this energy comes from. That will be cellular respiration. This is actually a glycolytic reaction, so which can run independently of electron transport chain. So that's not the answer. I think the energy is created from forming the bonds, as that gives off energy. Okay, so that's an interesting guess. This one I would venture to guess that 1,3-bisphosphoglycerate is higher energy molecule than 3-phosphoglycerate, so the phosphate group leaving is favorable. That's an interesting answer. I think that hits along it. Let's see here. Phosphorylation, cleavage of phosphate from 1,3-bisphosphoglycerate, okay, comes from the joining of the bonds and energy released by adding phosphoglycerate groups. Okay, so, we can look through this. I think that there are several answers. Here's one: phosphate makes a good leaving group since it creates a resonance stabilized structure. Okay, that's an interesting answer, and I think a fairly accurate one. So let's go ahead and do this. And then I noticed one up here: redox reaction. Okay, that's an interesting answer too. So let me go ahead and go here. Got to take my keyboard off. There we go. And let's just first of all recognize something here, all right, in terms of the reaction. So we have this molecule 1,3-bisphosphoglycerate, 1,3-BPG, that is being used at the outset of reaction 7 to catalyze the production of ATP. That molecule, if you were to look at this in terms of just dephosphorlating it, it will make 3-phosphoglycerate and inorganic phosphate, this PI. And if you were to look at the standard for energy change of that dephosphorylation event, it's going to be somewhere around negative 49 kilojoules per mole. Okay, so this is that's obviously a very exergonic reaction, and this 1,3-bisphosphoglycerate can be called one of these quote-unquote high energy molecules, that the removal of this phosphate is quite favorable from the first carbon of 1,3-bisphosphoglycerate. And it turns out that yes, indeed, the inorganic phosphate is actually a very good leaving group. Right, so in fact we could even say this, right, we could put this right in the category of fire molecules, right, and draw this like energy field around it, and that's the kind of molecule that we're working with. And again, we don't want to give magical descriptions of this, but the main reason this is the case is because that phosphate is a very good leaving group. Okay, and I don't want to get into the p-chemist definition of why that's a good leaving group and all that kind of stuff, although resonance does have something to do with it, which is what one of the answers mentioned. Right, now let's go ahead and do the opposite of this. If we can look at, let me get back to my black pen here. If we look at the production of ADP, again, you have to have an inorganic phosphate now being attached to ADP, okay, and this will make ATP. And the delta G of this reaction is about negative 30.5 kilojoules per mole. Not negative, positive, sorry. Negative is the hydrolysis, but to make it ATP, it costs 30.5 kilojoules of energy. Right? Now what you can see here is this, right? If we can couple these two reactions, there's enough energy in this process to drive this process down here. Okay, and that is indeed what happens. The enzyme phosphoglycerate kinase couples these reactions, and it takes the phosphate from 1,3-bisphosphoglycerate on the first carbon, right, and it uses ADP, the last phosphate in ADP, as a nucleophile to attack that inorganic phosphate, which is a very good leaving group, okay, as we've discussed, and that is going to then drive the synthesis of ATP. In fact, at standard conditions, again, this is standard conditions, not actual conditions, but this is about negative 18 kilojoules per mole. Okay, and so this is then favorable in the production or in the driving of the production of ATP. Okay, now what I want to go ahead and do is I want to take a step further into this and think about, okay, well, what about this molecule? Okay, how is that quote-unquote fire molecule or this high-energy compound, how is that going to be made? And that gets us back into reaction 6. So I'm going to come up here. Actually, I've already boxed it in here. But reaction 6 is catalyzed by this enzyme GAPDH, glyceraldehyde 3-phosphate dehydrogenase, and it makes from G3P 1,3-bisphosphoglycerate, right? And 1,3-bisphosphoglycerate is our quote-unquote high-energy molecule, right? So there must be something in this process that is conserving energy in that molecule, and there is indeed something in this reaction, and it's actually a redox reaction, right? And that redox reaction is energetically quite favorable, and it's storing its energy in the molecule 1,3-bisphosphoglycerate. Now, redox reactions are great ways to transfer energy. It's a transfer of electrons, if you remember from your general chemistry, but it's great ways to conserve energy or give off energy. In fact, when we think about our phones, redox reactions run our phones or our tablets or our computers, right? Laptops, when they have the battery, that is essentially carrying out a redox reaction between two half cells, and electrons are being moved from one half cell to another, and that's then driving the work that's being done by, of course, our tablets or our phones or anything else. Okay, well, biology does the same thing. It utilizes and harnesses redox reactions. In this case, though, we're looking at the reduction potential difference between a carbon and, let's say, a substrate, in this case glyceraldehyde 3-phosphate, and the reduction potential difference in a coenzyme, in this case NAD+, right? And that reduction potential difference is going to allow electrons to be driven from the carbon in glucose, so it was originally in glucose, now it's in glyceraldehyde 3-phosphate, to NAD+, and that is quite favorable because NAD+ has a higher affinity for electrons, and that then is going to be quite energetic, quite exergonic. Okay, and so we want to look at here this enzyme, glyceraldehyde 3-phosphate dehydrogenase, and how it will actually conserve energy in a redox reaction. Okay, now we're going to look at this mechanism. Okay, but understand something here. When we look at this carbon that's going to give up electrons, the electrons that are reducing that carbon or causing that carbon to be in a reduced state were originally derived from glucose. And that carbon was reduced, it was originally carbon dioxide and was reduced in the process of photosynthesis, and to do that, energy from the sun was required. We're not going to look at photosynthesis in this class, but that's where that energy got taken from and stored in that carbon that we're going to be looking at. Okay, so let's start here, and we'll just have our enzyme. Okay, obviously this is not a high resolution structure of the enzyme, but we have here the coenzyme NAD+ already bound, all right, and then we have a cysteine side chain in the active site. Okay, don't worry too much about these yellow boxes. Okay, I'm not going to spend a lot of time in these yellow boxes, but you can read about those on your own time. But let's focus on these other aspects of our enzymatic mechanism. Okay, so we move over here, going in this direction, and we form what would be considered the ES. Okay, and the ES is essentially the enzyme with the substrate bound, and here is our substrate, right? This is our substrate here, and that is G3P. Okay? Now, our next step, and really the very first step of the enzymatic catalysis, is the formation of this intermediate ES star. Okay? And that happens through this nucleophilic attack by this sulfur and this cysteine to this carbon in our substrate, and that's demonstrated by this arrow. Okay, so the arrow that I'm looking at here, I just highlighted in pink, but that's an arrow pointing to the arrow that represents our nucleophilic attack by the sulfhydryl group to that carbon. Okay, right, and that then produces this intermediate. Okay, and I've already underlined this intermediate, but it's called the thiohemiacetal intermediate, and that's denoted by this star, and that's what you're looking at here in the structure. Okay, and you'll notice here the main point would be that there's this covalent bond between the cysteine side chain and what was originally glyceraldehyde 3-phosphate. Okay. Now, no energy exchange has really happened. It's more of a setup step for this reaction to take place, right? But what's going to happen next, what's shown here in these arrows, let me go and highlight them. In particular, this one, okay, that is a representative of that redox reaction that I was referring to, okay? This hydrogen, along with the electrons in this covalent bond, are going to be used to reduce NAD+, all right? So NAD+ is our electron acceptor. The carbon, let me put a star by it here, that carbon is our electron donor. It's giving them electrons along with that hydrogen. It's actually transferred as a hydride anion. Right? That process, what you're looking at, is very energetically favorable. It's very exergonic, again, because of the redox potential difference between the carbon, the electron donor, and the NAD+, which is our electron acceptor. And that will make for a negative delta G. Now, certainly some of that energy is going to be lost as heat, but much of that energy is going to be stored in this next intermediate. And let me go ahead and highlight this for us. This intermediate, and I'm just going to highlight a portion of it, okay? This, and the portion that I highlighted is what's called the thioester bond. Okay? So thioester bond. And this thioester bond is a high free energy arrangement of electrons and atoms. Right? So it has a high potential energy, right? And it was formed, notice, right, by the redox reaction. In other words, the redox reaction allowed for the formation of this high potential energy bond, right, the thioester bond. Right? Now, we've got then energy stored in that bond. Notice now that NADH has been made, right? And we're going to go in this direction. Okay, NADH is going to be released, right? And we're going to bind inorganic phosphate here. This will be the inorganic phosphate that will eventually end up on ATP in reaction 7. But we have to place it onto our what was originally glyceraldehyde 3-phosphate, right? So that phosphate is going to come in, and it is going to act here. This oxygen is going to act as a nucleophile, and it's going to attack the thioester bond, okay? And what it's going to form here is an acyl phosphate bond. And that acyl phosphate bond is, like the thioester bond, very high in free energy, right? So what you see here is that we've got really an energetic, a very energetic reaction occurring all the way over here, the redox reaction, that then has its energy stored in the thioester bond, and then that energy is transferred to this acyl phosphate bond. And this then is how this, right, fire molecule, if you will, was made. Right, now that fire molecule then can come up here, going back to this, and can be used to drive the synthesis of ATP by essentially removing that inorganic phosphate, which is a very good leaving group. So there's a high potential energy, okay, in 1,3-bisphosphoglycerate, all right, and it's going to spontaneously drop to 3-phosphoglycerate and give up its phosphate to ADP, making ATP. Right, so I wanted to show you that, all right, and this will be one of the very few enzymatic mechanisms that we'll go through in this class, but I wanted to go ahead and show you that because one of the goals of this class is that you would understand how energy can be transferred from one place to another. So we've already talked about in previous lectures how, like, let's say an electrochemical gradient can be used to drive conformational changes in a protein that will then allow movement of some molecule against the gradient. So that's one way that energy can be transferred from, in that case, an electrochemical gradient to, to an enzyme or a transporter. In this case, we have a redox reaction that's transferring this energy to essentially chemical or bond potential energy that then is used to drive the synthesis of ATP. Right? So that's how energy can be moved around in these different pathways. Right? Now, the last thing that that we covered and got cut off in terms of the audio was these feeder pathways. And so let me go ahead and just highlight a couple of things here for us. We essentially looked at and have thought about glycolysis, and in that problem set you thought about glycolysis starting at glucose, and then that was converted by hexokinase to glucose 6-phosphate, and then on down the way it went. Okay, so this is essentially that prep phase up to glyceraldehyde 3-phosphate. Right? Now, it turns out, though, that in a multicellular organism such as you and I, or you don't even have to go that far, you could just go to even single-celled organisms, even prokaryotes, they're going to have, of course, other 6-carbon sugars available to them to absorb and utilize in the pathway glycolysis, okay, as are we. For instance, when you eat table sugar, that is a disaccharide comprised of both glucose and fructose. And so here on this slide are other types of 6-carbon sugars like galactose and mannose and fructose and how they can be used by the same pathway. And all this is showing is that these different enzymes or these different sugars will enter into the glycolytic pathway at some point in time in the prep phase. For instance, mannose over here, right, is converted to mannose 6-phosphate and then to fructose 6-phosphate. Okay, so it skips the glucose 6-phosphate and enters into glycolysis of fructose 6-phosphate. It uses hexokinase and phosphohexoisomerase, enzymes that I would think sound familiar to you, to get to that point. Galactose uses some kind of interesting pathways. There's several steps to make it this UDP galactose that is then converted to be UDP glucose, and then finally glucose 1-phosphate, and then finally that can be converted to glucose 6-phosphate. Okay, we'll actually encounter this enzyme when we get to glycogen metabolism. Okay, but so obviously that pathway for galactose is a little bit different than what you might consider for glucose or even mannose. Okay, now fructose can be used, okay, by hexokinase and converted to fructose 6-phosphate. However, hexokinase, the first step in glycolysis, has a pretty high Km for fructose, and quite honestly, fructose metabolism is not usually funneled through hexokinase. Rather, it's funneled through this pathway. Let me go ahead and highlight this here. Several different enzymes. You can look at those in your own time, okay? But eventually it will feed into glyceraldehyde 3-phosphate. Right? So that's just another way these 6-carbon sugars can enter into the pathway. Now, I do want to say one last thing here, in particular in regards to fructose metabolism, because fructose metabolism gets a lot of press. Okay, there's a lot of research indicating that fructose metabolism and diets high in fructose can lead to like metabolic disorders, including type 2 diabetes. And we're not going to get into all of that, but I do want to go ahead and look at this. So if you look at this here, this step is catalyzed by PFK1. All right? And of course, the steps above that are catalyzed by hexokinase. Well, hexokinase and PFK1 represent 2 of the 3 regulatory steps that regulate the flux through the glycolytic pathway. You'll notice here that fructose, its preferred route of metabolism, enters into the pathway below the regulatory steps. Right? So some of the theory in terms of why diets high in fructose might be unhealthy for us is because fructose enters into the glycolytic pathway below regulation steps, and so it's a less regulated metabolic route, and therefore could lead to problems, right? For instance, fructose, when it enters into the liver, it can increase rate through glycolysis by going this route, and if rate through glycolysis is increasing, then that's going to increase the rate of the production of pyruvate in the liver. What it's going to do to pyruvate is convert it to acetyl-CoA, especially in a fed state, and then acetyl-CoA is going to be converted to fatty acids. Those fatty acids will then be shipped out into the bloodstream, and one of the symptoms, okay, of a diet high in fructose is abnormalities in lipid metabolism. Okay, and that is essentially the start of that. Right? We'll probably talk more about those things later on. Okay, but for now, this will do it. Okay, so we'll come back and we'll continue on our discussion with glycolysis, in particular, start talking about shuttles. Okay.